Assalamu alaikum. Welcome to the Sunbar Technology Association. I am Sajid Ali Shah, and today we will discuss the role of CRISPR Cas9 technique and the treatment of HIV. So, in this video, we will discuss the HIV virus, its life cycle, the CRISPR Cas9 technology, and the role of CRISPR Cas9 technology and the treatment of HIV. So, about 39 million people were living with HIV at the end of 2022, and about 6 lakh 30 million people died due to HIV related illnesses worldwide in 2022. About 1.3 million people become newly infected with HIV virus in 2022, and currently in Pakistan, 1,65,000 people are living with the HIV virus. Different techniques have been used for treatment of HIV, but in the recent years, the three main nuclease-mediated gene editing tools, including transcription activator like nucleases, zinc finger nucleases, and clustered regularly interspaced short palindromic repeat CRISPR-associated nucleases Cas9, have been widely used in HIV AIDS treatments. Due to the costly and time-consuming constructions of terrains and the defense, the newest gene editing techniques, CRISPR-Cas9, has been developed. Human immunodeficiency virus. What is the human immunodeficiency virus? It's caused a disease which is known as AIDS. AIDS is a global health problem, mainly caused by HIV infection and induces immune destruction. There are mainly two types of HIV virus. One is HIV-1 and HIV-2. They both have similarities and both can lead to the development of AIDS. HIV-2 has lower transmissibility and is less pathogenic as compared with HIV-1, while HIV-1 is recognized as the major cause of AIDS. HIV-1 is a retrovirus with a genome of about 9.8 kb. The complete genome encodes 10 viral proteins including GAG, POL, VIF, VFR, VPU, ANV, TAT, REV, NEF, and the antisense proteins, as shown in the diagram number 1. Three main routes that spread HIV. HIV can be transmitted mainly by three routes, i.e., safe intravenous injections and vertical transmissions from parents to the upstream, i.e., from mother to their child through giving milk or during childbirth, etc. Structure of HIV-1 genome. So the HIV-1 genome is approximately 9.8 kb with 5 LTR, 3 LTR, and 3 LTRs, and it encodes 10 viral protein. It is the 5 LTR region, it is the 3 LTR regions. And these are the different proteins which were coded by these genes to perform different functions for the virus. Life cycle of HIV. HIV-1 invades host cell by binding its GP120 inverse proteins to the CD4 receptors on the membrane of the target cell and then interacts with the CCR5 or CXCR4 co-receptors. First, HIV binds through its GP120 proteins to the CD4 receptors on the host cell and then it interacts with the other co-receptors CCR5 or CXCR4s on the cell of the host. The host cells mainly correspond to T cells, monocytes, and dendritic cells, and even microglial cells, astrocytes, and perivascular macrophages and central nervous system. The life cycle of HIV-1 is complex, which contributes to ineffective virus eliminations, as shown in the figure number 2. The life cycle of HIV-1 provides possible CRISPR-Cas9 targets. The life cycle of HIV-1 completes mainly in 6 stages, i.e. binding and entry. The life cycle of HIV mainly completed in 6 stages. Number first, the binding and entry, second, reverse transcription, integration, replication, and assembly. And the fifth one is fourth is replication, assembly, and then binding. Figure number 2. It represents the life cycle of HIV virus. Firstly, HIV virus comes and binds to the CD4 receptors with the help of GP120 proteins on the cell wall of the host cells. After that, it interacts with the other co-receptors CCR5 or CXCR4 and fuses with the cell wall of the host cell, the cell membrane of the host cells and transfer genetic materials into the host cell. Here, it is the viral RNA. The viral RNA has been transferred into the host It is the best target for a CRISPR-Cas9 enzyme to degrade it here. Then reverse transcription takes place and viral RNA has been converted into the viral DNA. After that, it moves and integrated into the host DNA. This process is known as integration. After that, it's become the pro-viral DNA and it is transcribed and also translated into the viral proteins and also it's multiplicated and many copies of the viral RNA has been produced. After that, the assembly takes place. The viral RNA and the viral proteins combine and assemble to form the new virus, which moves to the cell membrane and is bud out from the cells as a new virus and then it moves and impacts the other cells in the body. HIV-1 HIV-1 entry into the cells will establish two types of infection. The HIV will cause mainly two types of infection. One is latent infection and the other one is active infection. For active infection, the provirus is active and produces viral particles which make infected cells but new progeny of virons. The establishment of latent infection may be mediated by complex mechanisms including RNA interference, chromatin environment, transcription factor, and HIV-1 provirus integration sites. The latent infection results in the generation of latent reservoirs contain infected which contains infected resting CD4 T cells, astrocytes, macrophages, and microbial cells. The latent reservoirs are often located in gastrointestinal tract. The latent reservoirs might mean the latent virus are being located in the gastrointestinal tract, brains, and lymphoid tissues, which are difficult for antiviral drugs to reach there and destroy them or degrade them and eliminate them from the body. Therefore, HIV-1 latent reservoirs are the major challenge for an effective cure for HIV. CRISPR Cas9 technology. It is a technology which was first found in bacteria. Bacteria use this technology as a defensive mechanism as an adaptive immunity against different viruses. CRISPR-Cas9, the CRISPR stand for clustered regularly interspersed short palindromic repeats, first observed in 1987. It acts as an adaptive immunity for certain viruses. The CRISPR-Cas9 apparatus involves the Cas9 helicases and single-guided RNA. The Cas9 nucleases has two actively domains, histidine, asparagine, histidine, HNH, and RUVC, cleaving different DNA strands. It is the target DNA strands and it is the case CRISPR-Cas9 systems. It is the guided RNA. The guided RNA moves and bind to the target sequence on the double-stranded DNA. Then the Cas9 has helicase activity, so it cleaves double-stranded DNA into two pieces, and then mutations take place there, i.e., addition of some nucleotide or substitution of some nucleotide or insertion of new nucleotide take place there. And in this way, the original strand of the DNA has been disturbed. It shows schematic diagram of HIV-1 provirus DNA modification by CRISPR-Cas9 technology. It shows CRISPR-Cas9 technology that how it make changes modifications in the DNA. So it is the provirus HIV-1 provirus double-stranded DNA. It is the guided RNA Cas9 system. It comes here and binds to the target sequence and cleavage is being produced here. After that, there will be two mechanisms. One is non-homologous engineering for the repairing of this DNA and the other is homologous dependent repair mechanism. So the one is in the non-homologous end joining mechanisms, there will be addition take place in either is the no template. No, here no template is being used here. So here maybe deletion take place, insertion of nucleotides may be take place, or some nucleotides will be replaced by other nucleotides. While in this case, there will be the need of a template, and then there will be the need of a template for the rejoining of the same strands again. So this is here the Cas9 enzyme has been used, while here in this case the DCAS9 enzyme has DCAS9 means the Cas9 enzymes have been critically deactivated. In this case, there are effector proteins which is binded with the Cas9 enzymes, and this technique is used for the activations of the latent virus in the cell. CRISPR-Cas9 technology application HIV AIDS treatment. Now we will study the applications of CRISPR-Cas9 technology and the treatment of HIV. So it is a highly active antiretroviral therapy. A CRISPR-Cas based approach was first tested in HIV/AIDS treatment in 2013. During 2013, recently the CRISPR-Cas9 system has been engineered as an effective gene editing technology with the potential to treat HIV-1 or AIDS. It can be used to target cellular co-receptors or HIV-1 genome to reduce HIV-1 infections and clear the provirus. It can also induce transcriptional activations of latent virus and the latent virus requires for elimination from the body. It has been successfully applied to HIV-1 or AIDS prevention and reduction in human cells and animal models. So, inactivation and elimination of HIV-1 provirus by the CRISPR-Cas9 technology. First, we will study that how it will inactivate and eliminate HIV provirus from the body. The main obstacles for HIV or AIDS HIV treatment is efficient targeting of the latent reservoirs. That we can't target the latent reservoirs efficiently. So, in early studies, researchers used three recombinases to specifically target HIV-1 LTR, which resulted in the excisions of HIV-1 provirus and HLA cells. A scientist, Q et al. Also generated ZFNs to target TAR region in HIV-1 LTR, which lead to the efficient cleavage of the integrated HIV proviral DNA in the core T cells and latent cell line C11. Epina et al., another study, used CRISPR-Cas9 to suppress the expression of HIV-1 genes in the core cell line by targeting HIV-1 LTR. The target sites were the NFK beta binding cascades located in the U3 region of LTR and the TAR sequences in the R region respectively. This resulted in efficient inhibition of HIV provirus transcription and replications. So another scientist study showed that the CRISPR-Cas9 could eliminate internal integrated viral genes from the infected host cells chromosomes. They used Cas9 guided RNA to target conserved sites in HIV-1 LTR regions, resulting in inactivating viral gene expression and restricting virus replication in HIV-1 latently infected T cell lines, promonocytic cell lines, and microbial cell lines. Targeting multiple sites of HIV-1 genome could increase the efficiency of excisions and disruptions of non-integrated proviral genomes. It means that when we target multiple sites in HIV-1 genomes, so it increases the efficacy of the excisions and disruptions of the non-integrated proviral genomes. Combinations of two effective sgRNAs to target different regions of the HIV-1 genome could prevent viral replication and escape. When we use two effective sgRNAs at the same time to target different regions in HIV genome, so it will prevent the replications and escape of the virus from the treatment strategy. CRISPR-Cas9 could cleave the non-integrated HIV-1, resulting in a 3 to 4-fold reductions of integrated HIV-1 provirus. The CRISPR-Cas9 functions in both HIV-1 and proviral DNA in latent cells, which make it more potential and promising HIV-1 AIDS treatment. Excision of HIV-1 proviral DNA in animal models was reported in 2017. It is the Cas9 gRNA attacked of the HIV DNA. That's how the CRISPR-Cas9 techniques attack on the HIV DNA. It is the double-stranded HIV DNA. It is the Cas9 system. So the guided RNA comes and binds to the target sequence, and then the Cas9 enzyme cleave it into two pieces. So now here maybe in the cleavage sites, maybe deletion takes place, some nucleotide may be deleted, or some new nucleotide may be inserted, or some nucleotide may be replaced by other nucleotide, and this is known as substitution. So here mutations will be occur in the viral RNA or the HIV DNA. The mutation may be deleterious to the HIV, and it may lead toward the cure of the HIV disease, maybe its replication stop, etc. Or mutations may be not deleterious to HIV, and in this case, the virus will be escaped from the treatment strategy. So next we will discuss. So these are the different targeted sequences on the HIV genomes which we were targeted through the CRISPR-Cas9 technology to eliminate them from the body. So SPCas9 enzymes will be used for this process, and this will be the delivery systems, and these are the targeting sequences. We will target these sequences. These are the targeted sequences, and this is their locations. And it show the efficacy that either when we target which one sequence to how it will be efficient for that the virus will be eliminated from the body. Now, the second strategy will be the disruptions of the co-receptor CCR5 and CXCR4 by CRISPR-Cas9 technology. So CRISPR-Cas9 technology can also be used to block HIV-1 entry by editing of the co-receptors. HIV-1 enters host cells through bindings to the CD4 receptors and CCR5 or CXCR4 co-receptors. It has been reported that individuals with a homozygous 32 base pair deletions of the CCR5 genes could live healthy and resistant or trophic HIV-1 infection naturally. Transplantations of CCR5 O32 HSPCs to a Berlin patient who experienced with acute myeloid leukemia and HIV-1 infection accidentally prevented HIV-1 replication and rebound so far. This is an encouraging case, but due to the few homozygous CCR032 donors, broad application of this strategy to HIV/AIDS patients is significantly limited. Even the CCR5 receptors are present in heterozygous conditions, then these patients will be resistant to the HIV infection. Gene editing of CCR5 by the defense and has been successfully used in CD4 T cells and CD34 HSPCs which could resist HIV-1 infection. Tevas et al. Conducted disruption of CCR5 via the defense delivered by adenoviral vectors and CD4 T cells from HIV patients and reinfused autologously after ex vivo extensions, leading to significant decrease of HIV DNA and RNA to undetectable levels in the host patient. But the other co-receptor, CXCR4, could also be modified by the defense to resist to export trophic HIV-1 infections. Compared with the defense technology, the CRISPR-Cas9 approach provides appropriate target sites with simple design and plasmid constructions. CRISPR-Cas9 has been widely used in disrupting CCR5 and CXCR4 expressions. In 2013, Chow et al. Showed that silencing of CCR5 by CRISPR-Cas9 can be successfully achieved in human embryonic kidney 293 T cells by transfecting Cas9 and sgRNAs. In 2014, Yi et al. Conducted a combined CRISPR-Cas9 or lens with the FAGI-VAC technology to perform the homozygous CCR5 or 32 mutations and induced pluripotent stem cells, iPSCs. These CCR5 modified iPSCs could normally differentiate into monocytes, macrophages, were, which were resistant to HIV-1 infections. In 2015, Lee et al. Used adenovirus delivered CRISPR-Cas9 combined with sgRNA which targeting the fourth exon of CCR5 to disrupt the CCR5 expression. Two specific sgRNAs can induce more than 60% cutting efficiency and TZM-BL cells. The results were also confirmed in Chinese hamster ovary and human T cell lines. Finally, they used a chimeric 85 F35 adenovirus vector to deliver the CRISPR Cas9 systems to human CD4 T cells to silence CCR5 expressions, which protected cells from HIV-1 infections with high efficiency and little off-target effects. How it all efficiently disrupted CXCR4 expressions and human CD4 T cells and races making CD4 plus T cells using CRISPR-Cas9 delivered by lentivirus and two sgRNAs. Other researchers also disrupted CXCR4 expression by Cas9 gRNA ribonucleoproteins Cas9 RNPs in human CD4 T cells, which reduced about 40% of CXCR4 expressions on the cell surface. So it is a schematic breakdown of gene editing strategies targeting CCR5 or CXCR4. So it is the CD4 T cells, it is CCR5 and CXCR4 receptors. So the virus binds here and the T cells death will be take place and the cell will be destroyed. Here the CRISPR, it is CCR5 locus in DNA, it is the guided RNA which target the CCR5 or CXCR4 receptor genes for Cas9, and it is the Cas9 the defense or killing enzymes strategy. So genes editing enzymes will be produced packed into adenovirus or lenti vectors which transfer these systems enzymes into the host cells. It is the CD4 T cells on which the CCR532 are deleted through the CRISPR-Cas9 systems. Now this cell is resistant to virus, and it is the CD4 T cells on which 6CR4 receptors, co-receptors have been deleted, mean their genes have been deleted, so no expression of this protein take place on the surface of the cells, and the cells become resistant to the virus. So now these are the some sequences which we can be targeted by the disruption of the CCR4 or CCR5 or CXCR4 receptors. So here in this case, the CRISPR enzyme used is SPCas9s. These will be the delivery systems. It will be the target genes. So we'll be targeted the CCR5 or CXCR4 genes in the body, and it is the strategy which strategy will be used for disrupting of these receptors, and these are the target sequences which will be targeted by the guided RNAs to move and bind to these sequences and changes and make cleavage in them and modification and make modified these sequences in order that they can't produce CCR5 or CXCR4 receptor co-receptors proteins in the body, and it is their efficiency percentage that either if we target which one sequence to how efficient will be our process. Now the other step is the reactivations of the latent HIV-1 virus by CRISPR-Cas9 technology. In this case, we will reactivate the latent virus in the body to reactivate them in order that we can target them through the drugs, or our body immune systems can eliminate them from the body. For this purpose, firstly we have to reactivate the latent virus in the body. So it is a potential tool for the activations of the latent HIV-1 viral reservoirs. It is also known as shock and kill strategy. It means that first we give shock to them, i.e. we activate them, and then we kill them through different types of drugs. To eradicate latent HIV viral reservoirs, it is necessary to reactivate the dormant virus and the host cells and induce cell killing by HAART and activations of antiviral immune responses. So histone D is different sustainable enzymes have been used for this purpose. The first one is histone deacetylase inhibitors will be used. They catalytically inactive DKS9s fused with transcription activator domains can activate viral gene expression in HIV-1 latent reservoirs, which may improve the shock and kill strategy. Zhang et al. Designed 20 sgRNAs to target the LTRU3 regions of the HIV-1 promoters and screened two target sites located near or at the NFK beta binding sites with high specificity and efficiency. This process leads to the reactivation of HIV-1 provirus in the HIV-1 latent cell lines such as TZM, BR1, epithelial cells, CHME5, microbial cells, and Jacquard T lymphocytic cells, induces suicide, death, and CHME5 microbial cells and Jacquard T lymphocyte cells. Another scientist designed 23 single SG guided RNAs to target the LTRU3 region of the HIV-1 virus and found the robust activation sites also near the NF Kava B binding sequences. Latency reservoiring agents, including prostatin and SAHA with CRISPR activators could increase latent HIV-1 reactivation. We can also increase the reactivation of the latent HIV-1 virus by using the different latency reversing agents, i.e., prostatin and SAHA. So, several proteins functions as restrictions factor during infection by HIV-1 and other viruses. So, simultaneous activations of the expression of restriction factors may represent an alternative strategy to prevent HIV-1 replications. Borgret et al. Used Cas9 based approach to induce the expression of the restriction factors A, B, FEOBEC3G, A3G, and FEOBFC3B, A3B in human cells. They also found that using two sgRNAs had higher efficiency than single sgRNA, and both activated proteins could block VIF deficient HIV-1 infection by inducing DC residue to DU residue. DC will change converted into DU, editing of HIV-1 gene. Recently discovered restriction factors such as serine incorporator 5, serine 5, human silencing hub, HUSH, and NONO may be new targets to be considered for these applications. SERINC5 inhibits virus infection by preventing virus and cell fusion. Activation of host restriction factors by CRISPR Cas9 is usually delivered by lentivirus with high transduction efficiency and virus productions. In addition, specific sgRNAs are essentials for the activations of target genes. Two sgRNAs will enhance when we use two sgRNAs instead of one sgRNA. So it will enhance the expressions of the target genes by increasing the target specificity to assure the complete activations of the promoter gene or a gene promoter. Limitation of the CRISPR-Cas9 application. So what are the limitations of this technology that we use this technology for the treatment of HIV? The first limitation is the one major concern as the potential of target effects. The potential of target is the first limitation of this technology, which may induce important gene mutations, due to which mutations occur in different genes and it's become more pathogenic or difficult for us. Another major challenge in CRISPR-Cas9 application is how to effectively deliver this large complex and HIV-1 infected cells. Another limitation is that how we will deliver it this complex system into the target site, into the target cells, which is already infected by HIV-1 virus. So it is the second limitations. CRISPR-Cas9 is considered as a potential antiviral tool, but HIV-1 has evolved escape mechanism. As we study and the early slides, that there were escape mechanisms. In some cases, the mutations, such type mutations take place due to CRISPR-Cas9 technology that the virus escapes from the treatment strategy. Another one is the CCR5 deficiency increases the risk of symbiotic West Nile virus infection. When we remove or when we suppress the expression of CCR5 receptors on the host cell line, it will increase the symptomatic West Nile virus infections. So it is another limitation of the CRISPR-Cas9 application. Now, what will be the future perspective of this study? So, exploration of new vehicles to deliver CRISPR-Cas9 compounds safely and effectively. We have to develop such types of vehicles, such types of transfer mechanisms for the CRISPR-Cas9 that we can deliver it effectively and safely to the target sequence. Another is by activating the latent viral reservoirs by CRISPR-Cas9, several specific agents can be combined to enhance immune responses to eliminate the virus. Third one is design specific sgRNAs and explore new strategies to decrease off-target effects. The another one is understanding the exact mechanism of viral escape from the CRISPR-Cas9 induced effects as critical for the designs of more effective strategies. We have to understand the exact mechanism that how the virus escape through this technique from the treatment strategy. The other one is the last one is the optimizations of animal models, HIV, one or else. First, we have to optimize this process on the animals model. Conclusion. Currently, in the conclusion, we will discuss a few points. So currently, HAART is still the major strategy for the treatment of HIV/AIDS patients in the clinics. Recently, broadly neutralized antibodies showed promising results. ZFN, by the size of approximately 1 kb, is earlier is easier to deliver. Nevertheless, the limitations of target site and high off-target effects make it difficult to be applied in HIV/AIDS gene therapy field. Talent is more flexible in DNA target design and has lower off-target effects compared with the defense. For CRISPR-Cas9, with more convenient and efficient design of target sites, less laborious vector constructions, limited off-target effects, it can be applied quickly in every research field, not only in HIV/AIDS therapy. For different target DNA, it needs only a change of sgRNA to find the most effective site, even though the large size of SPCase9 approximately equal to 4.1 kb decreases the efficiency of delivery. So the alternative is a Cas9 which has 3.3 kb size will overcome this limitation up to some extent. Thank you so much.